Another day, another story. Welcome back, fellow science enthusiasts. Today, we have an exciting episode lined up for you. We'll be diving into the world of bioassays, specifically focusing on how they help us understand the effects of various drugs. So, let's get started. Introduction to Bioassays. Before we delve into drug bioassays, let's begin with a brief introduction to bioassays themselves. Bioassays are experiments conducted on living organisms or tissues to measure the biological activity of a substance. They are an integral part of drug discovery and development, allowing scientists to assess the potency, efficacy, and safety of different compounds. Common types of bioassays. There are several types of bioassays used to evaluate drugs, but today, we'll discuss three common ones: receptor binding assays, cell-based assays, and animal model assays. Each approach provides unique insights into the effects of drugs. Receptor binding assays. Receptor binding assays involve studying the interaction between a drug and its target receptor. By using radiolabeled ligands or fluorescent tags, scientists can quantify the binding affinity and selectivity of a drug for specific receptors. This information is crucial in understanding how drugs interact with the body at a molecular level. Cell-based assays. Cell-based assays are performed using cultured cells that mimic different physiological systems in the body. These assays help researchers assess the drug's impact on cellular functions, such as proliferation, viability, and gene expression. By observing how drugs affect these processes, scientists can gain insights into their potential therapeutic uses or side effects. Animal model assays. Animal model assays involve testing drugs on living animals to evaluate their effects in a whole organism. These experiments help researchers understand how drugs interact with complex biological systems and how they may impact organ function, behavior, or overall health. Animal models provide valuable information before moving on to human clinical trials. Case study: anti-cancer drug. Now, let's walk through a case study to see how bioassays contribute to drug development. Imagine a team of researchers developing a new anti-cancer drug. They would begin with receptor binding assays to determine its affinity for specific cancer cell receptors. Explaining results after confirming the drug's promising binding affinity, the researchers would move on to cell-based assays. They would expose cancer cells to different concentrations of the drug to study its impact on their growth, apoptosis, and genetic markers. By carefully analyzing these results, the researchers can identify the drug's effectiveness against cancer cells. Transition to animal models. With positive results from cell-based assays, the drug would progress to animal model testing. In this stage, the researchers would administer the drug to tumor-bearing animals and monitor its impact on tumor growth, overall health, and potential side effects. Researchers' observations. Throughout the animal model assays, researchers would collect data on tumor size, metastasis, and any adverse effects observed. This information allows them to fine-tune the drug's dosage, frequency, and potential combination therapies. Conclusion. And there you have it. Bioassays play a crucial role in understanding the effects of various drugs. They help researchers evaluate a drug's potency, efficacy, and safety before progressing to human trials. By utilizing receptor binding assays, cell-based assays, and animal model assays, scientists gain valuable insights into how drugs interact with the body and their potential therapeutic applications. That's all for today's episode. I hope you enjoyed this journey into the world of bioassays. Remember, science is a fascinating realm, and bioassays are one of the pillars shaping our understanding of drugs. Stay curious, and we'll catch you in the next video. Until then, keep exploring and keep learning. Outro. Background music fades out. Thanks for watching.